All right, everybody, we are going to move on to chapter 16, which is the vascular system. I'm sorry if you can hear my dog chewing his bone next to me, but that's the only way I can keep him quiet right now. So we've already talked about the blood and the blood components itself. We've talked about the heart, or our main motor muscle and pump. So now we're going to talk about the rest of the vascular system, or really our peripheral vascular system. There it goes. Okay. So this is obviously quite a simplified version of what happens, but it's a good way to visualize it. So remember, arteries carry blood away. Arteries away. That's the easiest to remember. And then veins are the opposite. So veins are going to return blood to the heart. Remember, we talked about and we'll get to the lung circulation again, but we talked about in our heart chapter that the pulmonary arteries are the only arteries that carry deoxygenated blood, and the pulmonary veins are the only veins that carry oxygenated blood. But they still follow the rule of arteries carry away and veins return. And then we're introducing capillaries as well. These are the smallest arteries and smallest veins. So this is kind of our exchange point where we transition from arteries to veins. If you see, this is the right side of our heart. So we have our deoxygenated blood coming back through, in this case, the superior vena cava. You have our left side that has the oxygenated blood that's going to pump through the aorta. And there's a few other branches off the aorta, and that's how we get all the way throughout the body. And then as our arteries start to transition, we get to our capillaries. A lot of exchange of gases, nutrients, all that stuff happens in the capillaries. And then once that happens, they then kind of carry the blood that has lost its nutrients, oxygen, all that stuff back to the heart. Okay. All right, so let's talk about blood vessel structure itself. This is kind of the busy slide with the information, and then we'll talk through it on this next slide that shows you the visual too. Okay, so our blood vessel structure, there's three different layers to our vessels. The tunica intima, that is our innermost layer. So intima, so think inside. It consists of simple squamous epithelium. And so it's going to be continuous with the endothelium that lines the heart. So very smooth surface that allows for less resistance to keep the blood flowing. We'll talk a little bit about how resistance plays a role in things. Ooh, whoa. And then this layer also is going to produce our chemicals that cause vasoconstriction or vasodilation. So we'll talk about that concept as well. All right, our middle layer is the tunica media, so think M middle. So this is going to be our muscle layer, smooth muscle and elastic tissue. So this is going to allow that change in diameter. So the tunica intima will release our chemicals that give the instructions to dilate or constrict, but it's really the tunica media that has the muscle to allow constriction and dilation. And so this smooth muscle is innervated by the autonomic nervous system, if you think back to that chapter. So it's not like we get a signal from our vessels and our brain goes, oh yeah, let me go ahead and vasoconstrict or vasodilate in a conscious way. That's something that's auto-regulated. And then the outermost layer is our tunica externa, so think external. This is our strong, flexible, connective tissue layer. So this is going to be our support and protection. And so in veins, this is the thickest of the three layers. And then in arteries, the tunica media is more of our thickest layer, and that just has to do with kind of their function. So our arteries are under a lot more pressure. They, like I said, we have the vasoconstriction, vasodilation. That's why we need that thick muscle layer. Whereas the tunica externa in our veins is more, or our veins are more of a carrying pipe, so we need that extra support to help carry it back to the heart. Okay, so let's talk through it quickly with the actual picture. So you'll see the tunica intima is a very thin layer on that inside there. And like I said, this is simple squamous epithelium. It's called endothelium because it's on the innermost portion here. And as we said, it's continuous with the lining of the heart. So this stops our blood from sticking. It's kind of a smooth layer so that the blood has nothing that it can catch up on. Then we have our muscle layer here. You can see it's smooth muscle cells. You can see the difference here. We have epithelial cells versus muscle cells. And then our tunica externa, that's our connective tissue, fibrous layer. And so that's just our support and protection. Okay, this is just an actual microscopic view of that. So you see the tunica externa, very fibrous. There's a lot of connective tissue there, just woven to support things. You have the tunica intima in here. That's super zoomed in because of how thin it is. And then you can also see where the actual muscle cells are in here. So don't worry about too much about memorizing this. You won't see a picture pop up on your test or anything, but I think it's cool to be able to see what it actually looks like in the body. Okay, so we're going to talk a little bit more about arteries first. So they have a few different categories. We have our conducting arteries. They're going to be the largest and closest to the heart. So your aorta is the prime example of your conducting arteries. So they're going to be elastic. They're under a ton of pressure. Think our heart is pumping really, really hard. So they really need to be able to handle that rush of blood into them. So they have that elasticity so that when they get that rush, they can expand and they're not under too much pressure. So off of our conducting arteries, we're going to have a distributing arteries. So these are going to go to specific organs, and they're very muscular. They have that really thick tunica media layer that we were just talking about. So some examples of distributing arteries are your brachial artery, your femoral artery, and your renal arteries. So as I said, they're going to specific organs or muscles. And then we have our arterioles. They're going to be the smallest version of arteries. So arterioles are going to be before we hit the capillaries, but they're much smaller than our conducting and distributing arteries. So they have a lot of resistance to them because of how tiny they are, and they're going to control the blood flow into an organ. So kind of diving a little deeper, even when you get to the arterioles, you have meta-arterioles, and that's actually when they're connecting an artery or an arteriole to a capillary. Okay, so we're going to talk just a little bit about the difference in veins and arteries outside of the fact that one's carrying blood away and one's carrying blood back to the heart. So vein walls are thinner because they're not under near as high pressure like our arteries. They do have a good stretch ability, so they can carry a large amount of blood. So we also can refer to veins as capacitance vessels. So they have a large capacity to carry blood. Veins can constrict extensively. So I talked about vasodilation and vasoconstriction in terms of arteries and that tunica media layer, but veins can also constrict a lot. They just don't have as much muscle to do it because they don't have as much pressure that they're fighting against. And in general, they're much closer to the surface of our skin than our arteries are. Okay, so we're going to go kind of the opposite order. So we went conducting arteries, distributing arteries, arterioles. Like I said, then we would hit capillaries. And now we're going to be on the other side of the capillaries. We're going to have venules. They're very small, thin, porous walls, meaning that it's easy for exchange to happen there. We have medium-sized veins, which is really kind of venules coming to join together. Think the roads are merging onto the highway. And they're going to contain one-way valves. So we'll talk about this a little bit kind of like towards the end of this lecture. But because our veins are fighting gravity somewhat to bring things back, we don't want backflow. Just like the valves in our heart try to prevent backflow from chamber to chamber, we don't want blood that we're trying to return to the heart going back the wrong direction. So those valves kind of open up as we pump one way, and then they close to make sure that that blood doesn't fall back down. And then finally, we have large veins. They're formed by, again, I think we have the the venules merging into medium-sized veins, and then we have our medium-sized veins forming into our large veins. So they kind of just meet and meet and meet, and eventually we get one big vena cava to take it back to the heart. And so they have that really thick tunica externa because they have a large amount of volume of blood. They need that extra support to maintain shape and all that good stuff. Okay, so our capillaries, these are our exchange vessels. So we need these to actually have the exchange occur itself. So they have very thin walls, very small diameter. As I said, they link our arterioles to our venules. And so they're going to be really abundant in tissues that have a higher metabolic rate because in those tissues, we need a lot more exchange to happen. So you can see it in the visual down here. They have very small diameters, barely wide enough for blood cells to pass. And they're only made up of that endothelium, which has a basement membrane, and then a really thin wall so that it's easy for substances to come and go. Okay, so our capillaries are organized into networks known as capillary beds. So it's not like one capillary connects one arteriole, one venule. You'll see if we go back up, this would be your capillary bed, kind of that whole network of capillaries. All right, so they form the microcirculations by attaching the arterioles and venules. That's another word for that capillary network. And then I want to highlight one special kind of capillary known as a sinusoid capillary. And so it is a special type of capillary found in some of the organs. The liver is an important one. The spleen is another important one. And these are also found in bone marrow. And so we already talked about capillaries being quite permeable, but these sinusoid capillaries are even more permeable than normal. So larger substances such as proteins, new blood cells from the bone marrow, that kind of stuff, we can actually get past the capillaries, which would not normally be able to pass in a normal capillary. And as I said, so for our bone marrow example, this is how we can get new blood cells to enter the bloodstream. The spleen is like a massive store of blood cells that kind of helps regulate our blood volume, so they can get more blood into the bloodstream that way as well. So remember, sinusoid capillaries are special type. We only find them in some places, and they're very permeable. Okay, so the actual organization of the capillaries, it's how we kind of make sure that we get blood where we need to, and we don't want to be inefficient with the use of our blood. So this is picture on the left is showing during exercise. When we exercise, a lot of our muscles are going to need more blood flow. So as that blood travels through the arteriole, these precapillary sphincters are going to open. When they open, we can get blood through all the different arterial capillaries. They're going to flow into the venous capillaries. They're going to come together and meet at the venule. So that's with exercise. You see all of them are being utilized. With rest, we may close off a good portion of those capillary sphincters and have just one pathway that's going. So like I said, we don't want to waste the use of our oxygen, nutrients, all the stuff that's traveling with the blood. Okay, we're going to go into capillary exchange itself. So the walls of the capillary, we said, are quite thin, but they allow for a two-way exchange. We're going to release chemicals such as oxygen, glucose, the stuff that our tissues need are going to go out of the capillaries. And then waste such as carbon dioxide, ammonia, a good amount of substances are going to go in so that they can pass to the veins. So we're going to talk about a few different mechanisms of this exchange, including diffusion, filtration, and osmosis. But I'm going to go ahead and take a pause so that my laptop doesn't die on you guys. And we will pick up here with a part 2.